Okay, hi everybody. My name's Mara Carroll. I'm the Director of Development here at Jett Foundation. Thank you all for joining our community webinar series. Today we are welcoming Dyne Therapeutics to discuss their new clinical data from their DELIVER trial. We are grateful to our partners like Dyne Therapeutics for helping us make it possible to bring these webinars right into our families' homes. Before we get started, I did just want to mention a few things. This webinar is being recorded, so if you have to log off for any reason, it will be recorded and live in our library of resources on our website, and it is also being streamed live on Facebook. At the end of the webinar, we will be having a live Q&A, so if you have questions at all throughout the webinar, feel free to put them in the question box, but just know that we will answer all the questions at the end of the webinar. I think that is all from me. So without any further ado, we'd like to welcome Ash Dugar, Chief Medical Officer for Dyne Therapeutics, to present for Dyne. Take it away, Ash.
Oh, wonderful. Thank you, Maura. So as Maura said, I'm the Chief Medical Affairs Officer at Dyne Therapeutics. And first, I'd like to thank Eric, Maura, and the entire Jett Foundation for the opportunity to present our latest data release on our Duchenne program. It's always a privilege to speak to the Duchenne community, and I want to thank you for your time and attention today. With me today is our Head of Patient Advocacy, Catherine Beaverson. And she will be joining me again at the end of this to take some questions at the end of the talk. And I'm also joined here by Tiffany Thompson, our Medical Affairs Operations Lead at Dyne, and someone that also helped us make sure that we are ready for these webinars. So thank you to the Dyne team. Next slide, please. So I'd like to remind everyone that I will be making forward-looking statements today, and you can refer to our website for more information. Next slide, please. It's really important for me when I have these conversations to start with this slide, because at Dyne, it all begins with our mission, which is to deliver life-transforming therapies for people living with serious muscle diseases, with Duchenne being a priority. This mission is woven deeply into our core values and inspires our employees each and every day. Next slide, please. So there are three key challenges as to why currently available antisense oligonucleotides, specifically PMOs, or phosphorodiamidate morpholino oligomers, PMOs, have significant limitations in terms of dystrophin restoration and functional improvement. Now, these are exon-skipping PMOs designed to create truncated functional forms of dystrophin. But it's been quite challenging, largely because of these three challenges. The first is being able to get the PMO to the nucleus to take advantage of the cellular machinery that exists. So it's really getting the PMO not just into the muscle and into the cell, but into the nucleus of that muscle cell so it can attack the genetic basis of Duchenne. The second challenge is the targeted delivery to muscle fibers. Historically, the PMOs have not been able to get to all of the muscles that they need to get to, including cardiac muscle as well as the breathing muscles. And so that's become a huge challenge that many companies are trying to overcome, which is this targeted delivery of the PMO to muscle fibers and getting broad and deep distribution in those muscle fibers. And so that brings us to the third challenge, which is the broad distribution, like I just said, and to those muscles that matter. So getting it to the skeletal muscle, the breathing muscles, the cardiac muscle, the smooth muscle, meaning the gut, and even the brain. So really being able to get the antisense oligo, the PMO, into all of the muscles that matter in Duchenne, to have broad and deep distribution across muscle fibers. Next slide, please. So thinking about these challenges is what really has driven Dyne to develop its FORCE platform and specifically DYNE-251 as a next generation exon skipping investigational medicine. So the platform that I'm showing you here is expressly designed to efficiently and reliably deliver PMOs to those muscles that matter, the skeletal muscle, the cardiac muscle, and the smooth muscle, and target the genetic basis of disease. It consists of three components. On the left hand, on the top left hand of the slide here, you can see the Fab, or the antigen binding fragment, or the antibody fragment. We call it the Fab, which is the top portion of this depiction you see here, this Y-shaped figure of a full monoclonal antibody. So we took the top portion of that monoclonal antibody and engineered it to bind tightly to something called the transferrin receptor 1, or TFR1. Why did we choose the TFR1? We chose it because it's highly expressed across the surface of muscle cells, including the heart and breathing muscles and skeletal muscles. So the Fab targets this TFR1 and uses it to deliver the PMO to muscle while maintaining the ability of the TFR1 to do what it's designed for in nature, which is to transport iron into the cell. The second component is at the bottom here, the clinically validated linker. Now, we chose this because of its demonstrated safety and efficacy in third-party approved products. So this linker in the burnt orange string is actually used in products that are on the market in the US for oncology. And this linker enables precise connection of the Fab to the PMO, the exon skipping PMO. And finally, here you can see in this, this puzzle piece, an exon skipping PMO, which we rationally selected to target the genetic basis of disease. And this is where we can leverage the modularity of the FORCE platform. And what I mean by that is for nuclear targeting diseases such as Duchenne, we choose antisense oligopayloads. And in this case, we chose an exon skipping PMO that targets the specific mutation in the DMD gene. And DYNE-251 is our investigational medicine that uses an exon skipping PMO designed to enable skipping of exon 51 to make shortened functional dystrophin. Now, the FORCE platform is designed with the goal of addressing the current limitations of delivery to muscle tissue and to improve the delivery of the PMO to those muscles that matter that I just mentioned, the cardiac muscle, skeletal muscle, including diaphragm. And we also believe this modularity and what we can deliver with this platform will allow for increased time between doses. And that's going to be really important for us to be able to provide a medicine, a potential medicine that is as helpful to families as possible. Because right now, current PMOs require weekly dosing. And so these next generation exon skipping drugs, such as ours, are really exploring more infrequent dosing, which I think is quite important. Next slide, please. The mechanism of FORCE is different from other approaches, and we often get this as one of the first questions when we're talking to the community. As I mentioned before, we're essentially using the cell's natural biology to deliver our investigational medicine to muscle cells. Our Fab binds tightly to TFR1, as I said before, and we use the TFR1 again because it's highly expressed across the surface of muscle cells. So the Fab acts as a kind of key that unlocks the TFR1 receptor and allows our PMO to enter skeletal, cardiac, and smooth muscle to target the genetic basis of disease at the level of the nucleus. So in a way, we use a Trojan horse approach to drive the PMO into the cell using the TFR1 receptor without disrupting its normal function of iron transport. So the FORCE platform harnesses the natural mechanism of the TFR1 receptor mediated delivery to transport the PMO across the cell membrane. And this is differentiating because we are not using membrane destabilizing agents to deliver the PMO into the cell or leave the endosome to travel into the nucleus. And by targeting muscle specifically by using TFR1, we have the potential for a wide therapeutic or safety window, resulting in less frequent dosing and chronic administration. Next slide, please. So today, Dyne is well on its way towards achieving the promise of FORCE and delivering for patients. We've delivered some really exciting clinical data, and I'm pleased to share that data with you today. Next slide, please. So just as a reminder, I wanted to make sure that we showed the amount of feedback we received from key opinion leaders and advocacy leaders and the community in designing our clinical trial. We engaged these KOLs from various disciplines, including pediatric and adult neurology, cardiology, physical medicine and rehabilitation, pulmonology, and physical therapy. And importantly, we've gathered this feedback from across the globe. Now, KOLs, both inside the US and outside of the US, with experience running early and late stage trials, have provided advice on our overall clinical development program, including the clinical trial design, the patient population that we're testing in DELIVER, the biomarker and functional endpoints we're evaluating, and key safety considerations. And importantly, they've provided key learnings from numerous long-term natural history studies to help inform our trial design and eventually to contextualize the results that we obtain from our trial. While it's clear from talking to KOLs that dystrophin is still a key endpoint for this condition, both the physicians and the patient community have been very clear. Function is what's most important. In designing the trial, we've also received advice directly from patients and caregivers who we consider as experts, certainly experts as well in the disease area. And we've also obtained advice from advocacy leaders from around the world, including the Duchenne Community Advisory Board, which comprises of leaders from several countries across the globe. They provided insights based on their living with and being the voice of the Duchenne community, as well as experience with past and current trials. And this input has been critical in ensuring a patient-centric design and has included, but it's not limited to, key considerations for choosing a clinical trial because there are so many choices now. Review of and input to our informed consent form to make sure that it's providing useful and necessary information to the community. Feedback on biopsies and functional outcomes, identification of the most burdensome or difficult parts of the trial, and how to ensure patients and families are supported throughout the process. Next slide, please. So before I go into detail on the data, this is a brief overview of the DELIVER study design. It's a phase 1/2 global trial that we've designed to be potentially registrational. This trial is currently enrolling ambulant and non-ambulant males with Duchenne who are ages 4 to 16 and have mutations amenable to exon 51 skipping therapy. Patients who've been previously treated with exon skipping therapy can enroll in DELIVER after a 12-week washout period. The primary endpoints are safety, tolerability, and change from baseline in dystrophin levels as measured by Western blot. Secondary endpoints include measures of muscle function, like the North Star Ambulatory Assessment and Time Function Test, all of which I'm sure this community is very well versed in, as well as exon skipping. I have to point out we also have wearables to allow collection of real-world data, such as stride velocity. And interestingly, this endpoint has been accepted by EMA, the major European regulatory authority, as a primary functional endpoint in Duchenne trials. For details on our inclusion and exclusion criteria, you can visit our ct.gov page for more information about the trial design, as well as the IE criteria and clinical trial sites. Next slide, please. So I'm going to start to go through the data, starting with the safety data and then moving to the biomarker data and then the functional data. So DELIVER, so on this slide is a summary of our safety profile. What we can see in this study so far is the safety profile has been favorable based on more than 50 patients enrolled in DELIVER with over 670 doses administered. So we have obtained a great deal of experience with this drug so far. The majority of our treatment emergent adverse events were mild or moderate in nature. It's important to point out there have been 3 serious treatment-emergent adverse events potentially related to study drug in 2 participants in the 40 milligram per kilogram PMO equivalent dose study group, which was the highest dose tested. Both participants are home and doing well. Other than those 2 participants with serious treatment-emergent adverse events in the 40 mg per kg cohort dosed every 4 weeks, no participants have demonstrated persistent related anemia or thrombocytopenia or demonstrated kidney injury. And no participants have demonstrated clinically meaningful changes in electrolytes, including magnesium, as seen with other programs. All patients in DELIVER are now being dosed at 20 milligrams per kilogram. So that includes participants dose escalated following the placebo-controlled period from the starting doses lower than 20 milligrams per kilogram, and the participants that initiated at 40 milligrams per kilogram who are now being dosed at 20 milligrams per kilogram following evaluation of the safety profile. So bottom line is that all the participants in DELIVER are now being dosed at 20 milligrams per kilogram PMO equivalent dose. Next slide, please. So first, I'm going to go through the orientation of the slide, and it's the same setup for the next few slides. The gray bar, which is just a sliver here at the bottom left, represents the placebo-treated patients. The blue bar are patients dosed at the 5 milligram per kilogram PMO dose. The teal is 10 milligrams per kilogram, and the dark blue is 20 milligrams per kilogram. This slide shows muscle concentration, and you can see that DYNE-251 drove dose-dependent increase in PMO muscle concentration. This confirms that the drug is getting into the muscles and more gets in with higher doses. Next slide, please. Similarly, we saw a dose-dependent increase in exon skipping. Here you can see that at 10 milligrams per kilogram, we saw nearly 2% exon skipping, and at 20 milligrams per kilogram, around 3.3% exon skipping. So we were very pleased to see this level of exon skipping at these doses. Next slide, please. So we're particularly enthusiastic about the dystrophin data as measured by Western blot. Here you can see that at the 10 milligram per kilogram dose delivered every 4 weeks, DYNE-251 reached a 3.2% level of dystrophin, about a 3% change from baseline. And at 20 milligrams per kilogram, we saw an absolute dystrophin level of about 3.7% at 6 months, and that's about a 3.15% change from baseline to the 6-month period. So we see that DYNE-251 continues to drive that robust dystrophin expression in these cohorts. Now, the dystrophin analysis here is unadjusted for muscle content, and we believe this is an appropriate evaluation when looking at dystrophin, and we know that there are many numbers that are put out there from many companies, but it's really important to understand the change from baseline to be able to understand what kind of treatment effect we're having. And, from our perspective, these data are quite striking. Next slide, please. However, we do know that others in the field have adjusted for muscle content. So we've done this analysis as well. And here you can clearly see how we've calculated this. The calculation is on the slide here. So we're trying to be transparent about how we are conducting this adjustment for muscle content. And what you can see here is when we make this adjustment in the 10 milligram per kilogram cohort, from baseline to 6 months, there's about a 7% change in dystrophin. And in the 20 milligram per kilogram cohort, the change from baseline is about 7.4% with an absolute of 8.72%. So we wanted to be very transparent and share the full set of data that we've generated and also provided a view of what the muscle content adjusted dystrophin amounts to be. Next slide, please. So what's really exciting is that we see this robust dystrophin expression lead to encouraging trends across multiple functional endpoints, across multiple doses at just 6 months. Here we see stabilization or improvement with 10 milligrams per kilogram of DYNE-251, while participants receiving placebo declined over that same period of time. And we can see here in the teal line, this sustained improvement in function at these various endpoints. You can see here at day 169, and then again at day 337, improvement or stabilization across the NSAA, the time function tests, such as time to rise from floor and 10-meter walk run, and very importantly, in the stride velocity 95th centile. Again, this is a measure in the real world setting using a passive assessment. So it really has not the biases that some of the other functional endpoints have. In the black line, you can see the 20 milligram per kilogram cohort at 6 months. And in just this short period of time, at 20 milligrams per kilogram, we are seeing an improvement in the NSAA total score, in the time to rise from floor, and stabilization in 10-meter walk run, and then importantly, improvement in the stride velocity 95th centile. And the other thing that excites us about the 20 milligram per kilogram cohort data is that at baseline, these participants were much more impaired than the participants in the 10 milligram per kilogram cohort. So this group was older and had a lower baseline functional status than the 10 milligram per kilogram cohort. And despite that, we're able to see robust improvement across multiple functional endpoints. Next slide, please. I just wanted to double click a little bit on the stride velocity for those who may not be so familiar with this particular endpoint. So as I mentioned before, it is qualified to be a primary endpoint by EMA in studies on boys who have Duchenne who are greater than or equal to 4 years of age. This is a digital endpoint. It's a wearable, objective endpoint of ambulatory performance in patients' normal daily environment. So they don't need to come into the clinic. They take this device home, they wear it at home, and they conduct their lives, without any intervention. They are just doing what they do, and we're collecting the data with this device. Importantly, this outcomes measure is sensitive to detect change over a short period of time, which has been shown in natural history, steroid-treated patients, and in clinical trials. It is correlated with traditional clinic-based outcomes, such as 6-minute walk test and North Star ambulatory assessment. The validation of this endpoint has been done very rigorously and importantly to identify a minimally clinically important difference. So what is actually clinically meaningful? That change has been defined and accepted by clinicians and by EMA. And so if we go to the next slide, this is that one graph just enlarged. And again, I want to point out that at the 10 milligram per kilogram dose, we are seeing an improvement that is above the minimally clinically important difference established in various studies. The 20 milligram per kilogram dose at just 6 months is also showing improvement in the stride velocity 95th centile. And one can see here that separation from placebo in the gray bar, which is declining, similar to what you would see in natural history. And so we're seeing not only improvement, but a large separation from placebo. Next slide, please. So in summary, what we've tried to communicate today is that patients treated with the 20 milligram per kilogram dose every 4 weeks had a mean absolute dystrophin level of 3.7% and 8.7 when adjusted for muscle content. We're really excited to see improvements, trends for improvement or stabilization in multiple functional endpoints, at the 10 and 20 milligram doses with respect to baseline to 6 months, and for 10 milligrams, even going further out to 12 months. And again, we've seen meaningful improvement in a real-world outcomes measure, the stride velocity 95th centile, in both the 10 and 20 milligram study groups following dosing every 4 weeks. Now we're initiating our registrational cohorts in the DELIVER trial, and we plan to provide an update on the path to registration by the end of 2024. Next slide, please. So with that, I would like to thank the Jett Foundation for the opportunity to present to you today. Thank you to the families, investigators, and sites that have made this and so many other advancements in Duchenne possible with your countless sacrifices and dedication. What the community's done over the years is nothing short of extraordinary. So thank you.
Great. Thank you so much, Ash. We really appreciate your thoughtful presentation and bringing that to our webinar series. And so now we would like to open it up for questions and answers. I think a few questions had come in with the registration that I can go ahead and ask, but also anybody that's listening here today, hopefully you got a lot of your questions already answered from the presentation, but feel free to put them into the question box, and Ash or Catherine will get answer for you. So this one question came in, and you've sort of just acknowledged this, but when will you start more cohorts? Will you open up more sites in Europe?
Yeah, great question. So I think there's a couple of questions, and I see one in the chat as well. What does it mean in terms of registrational cohorts? When are you opening up registrational cohorts? So, we believe we've engaged with a number of regulatory agencies around the world. As mentioned, we believe we're conducting a study that certainly based on precedent in the US is more than adequately designed for accelerated approval in the United States. And as I'm sure folks know, there are 4 exon skipping PMOs that have been approved in the United States through the accelerated approval pathway. So there's a very clear regulatory pathway, and so we feel very good about the design we've put forward. We are currently enrolling additional cohorts in the 20 milligram per kilogram dose cohort, PMO equivalent. And then beyond that, I'm happy to come back to the community when we talk about opening up even more cohorts, should we do that. So that's something, that I'd ask everybody to stay tuned on. But we are currently enrolling additional 20 milligram per kilogram cohorts to make sure that we bolster the amount of patients for both safety and efficacy.
Great, thank you. Any other questions? Anyone else? Please feel free to.
There was another question in the
Yeah.
chat around going younger than 4 years of age. So the DELIVER study, as I mentioned, has patients from 4 to 16 years of age. Again, we're following a similar path that's been accepted by the FDA. We do have, we have been talking about going to earlier ages. We firmly believe that the earlier, these boys can start, these individuals with Duchenne can start on exon skipping treatment, the better. The muscle damage occurs early on, even though symptoms may only show around the age of 2. So we believe that it's important to be able to treat patients as early as possible. So we're getting through our phase 1/2 study, our DELIVER study, and then we are thinking about how to evaluate participants who are younger than 4 years of age. I think it's critical for us, frankly, for every company, to be thinking about how to do that. And hopefully newborn screening, and maybe Catherine will have some comments, but hopefully newborn screening is on the horizon through the federal RUSP and then going state by state. But we think the time is absolutely right for all of these companies to think about programs that allow evaluation of kids that are, as young as 6 months, 3 months. Catherine, I don't know if you want to add to that.
No, I have nothing to add, but Ash, I love the question that's coming through on the chat regarding wearable technology.
Mhm.
Um,
Yeah, I can go ahead and read that. We just got a comment. I really like the wearable technology as an endpoint for the ambulatory population. Do you think similar technology would be available in non-ambulatory patient population in the future?
so I actually, I hope it's not in the future. I hope it's now. It's a great question. It's a really, really good question. So the side device, which is the device we're using to look at stride velocity 95th centile, it has much more function than just looking at that outcomes measure. So that actually can look at functionality in a non-ambulatory patient population. I keyed in on the stride velocity because that's the outcomes measure that's been validated by EMA and qualified as a primary endpoint and used in a couple of interventional studies, including ours. So it's the outcomes measure. But there are devices out there that already are looking to evaluate upper limb function, for example, in non-ambulatory patients, even sleep, cardiac function. So there are a number of wearables that are already available. Dyne is looking to implement other wearables in our DMD program. And so right now we're not ready to communicate which devices and which wearables we're going to look at, but we are looking to implement other wearables to measure function in both ambulatory as well as non-ambulatory participants.
And Ash, may I build on your answer? Because again, I think this is a fantastic question. And it's an add-on to the previous question as well. When we think about going into the younger Duchenne population, right? It's as we look to expand into older or non-ambulatory, I think we would love and welcome and will plan to engage with the patient community exactly to understand what can be measured, what should be measured, what means most to folks who are currently non-ambulatory.
Yeah. And I'm going to build on that for a second. I think it's a really good point. And just even more broadly, the DELIVER study, as I mentioned, was designed with input directly from the community. We published a paper, driven by Pat Furlong, in a that was published in a peer-reviewed journal that delineated exactly what we did to get that input into our DMD study as well as our myotonic study, and how we implemented that feedback. And the result is the study that I've presented to you today. So to Catherine's great point, as we think about what else we want to do in our clinical program for DYNE-251, we will continue to get feedback from the community to make sure that we're doing doing what makes sense for the community and for the physicians.
Great, thank you. The next question came through. Can you go for accelerated approval at EMA? Will you consider that?
A great question. So for those of you who may be familiar with EMA regulatory statutes, there is a pathway for expedited approvals with EMA for the European Union, for MHRA with the UK, even PMDA with Japan. So there are expedited approval pathways for ex-US regulatory agencies. And so we are absolutely thinking about how to leverage those expedited pathways to be able to get DYNE-251 to as many patients who need them as possible. So yes, we are thinking about how to leverage those expedited pathways for EMA as well as other jurisdictions outside of the US.
Great, thank you. There was another question that had come in with our registration questions. When will this be available to all kids that are amenable to exon skipping? I'm guessing maybe they're saying in addition to exon 51 if you have, additional exons in your pipeline. I'm thinking
Yes.
that's what we're asking.
Yeah, so I was remiss in not mentioning this at the end of the talk, so forgive me. So we are pursuing a broader set of exon skipping therapies. Again, this is around the modularity of the FORCE platform. So for this program, for the myotonic dystrophy program, for other programs, we use the same Fab and the same linker. And we interchange the antisense oligonucleotide that we're using if we're going after targets in the nucleus, such as for Duchenne. So this modularity enables a more efficient pathway in terms of preclinical and clinical development. So we are pursuing programs for 53, so for skip amenable, exon 53 skipping, exon 45 skipping, and exon 44 skipping, and even beyond that. Right now, the programs that we've talked about are those three programs. So we're advancing that in the preclinical space. At the same time, we are trying to be thoughtful about how to leverage the non-clinical and clinical data from our DYNE-251 program to make it more efficient in terms of the preclinical and clinical development for the follow-on exons. So again, we're pursuing exon skipping programs for 53, 45, 44, and also we are working with regulators about the most efficient way to move these programs forward. I don't think anybody wants to have 5 years per program. So we need to be able to work with the community, with physicians, and with regulators to find a more, efficient way to move additional DMD exon skipping programs forward. So we're doing everything we can towards that end.
Thank you. Another question asking, will you be opening more sites in Europe? I know you mentioned more to come at the end of 2024 and more announcements, but if you have any more to elaborate there.
So, we're always looking to be able to have more sites if it makes sense for the program. So there are reasons in terms of being able to make sure that we have, as we increase the sample size, we know there's going to be limitations because there are so few, patients around the world just in general. Plus, there's a number of other, programs that are also in process where families may choose to go. And so the good thing is, I think there's a lot more choices in the last several years in terms of Duchenne programs. I know it's a double-edged sword because then the decision-making becomes quite challenging as well. So we are exploring adding sites in our DYNE-251 program. And I would ask that you go to the ct.gov webpage or the EU Clinical Trials webpage for the full listing of sites. And as we bring on new sites, they are updated nearly automatically. So we're continuing to explore the best path and the best sites as we expand our program.
Great, thank you. Another question just came in. When will DELIVER study come to an end, and when can you ask for approval?
Yeah. So it's as I think as I communicated, it's a multi-year study. Right now it's a 3-year study. And so we have a 6-month double-blind placebo-controlled portion. And then we have everybody go from the 6-month period into an open label extension where everybody receives drug. And that's a 24-week open label extension, and then a 96-week long-term extension period. And as I mentioned, everybody is now at 20 milligrams per kilogram. So we're running this study out as designed and as agreed on with regulators. So I can't give you a precise answer as to when the DELIVER study will come to an end. We obviously have mapped it out. We have our own timelines. And if you go to ct.gov, you can see that we do have an estimated completion date for the DELIVER study. Now, I guess the other piece of this, which is part of the question, is there are, as I mentioned, those expedited approval pathways. So in the US, for example, there is there are examples, there's precedent for companies that have developed exon skipping drugs that are now approved. They have those programs ongoing, yet have extracted data to allow for accelerated approval using dystrophin as the biomarker that is likely to predict clinical benefit. So my point is, we don't have to get to the end of the 3 years for every patient in the study to ask for approval for the drug to be on the market. There is precedent in the US, and there are pathways in other regulatory jurisdictions.
Right. So clarification, you're 6 months in to sort of a 3-year study process. No.
No, sorry. No, I didn't mean it like that. I think we are, the 6-month portion is just the multiple ascending placebo-controlled portion. And so we are much further into the study than 6 months. We've been running it for multiple years now. But I was just saying, like, one can look at data cuts at different time points to decide if you have sufficient data set to go and talk to a regulator about an expedited approval prior to having every patient come, 3 years out of the study. We aren't giving what I would say, I guess where I'm dancing here is, as most companies, we're not giving specific guidance right now as to when we're going to go after, when we're going to go for approval. That's not something we're ready to communicate.
Absolutely. I know a lot of the questions are obviously all around urgency, and we are all excited.
We feel it. We feel it. We are.
You know it.
Yeah.
But this is very exciting data for you guys to be sharing today. So thank you so much. Ash and Catherine, I don't see any more questions. So again, for all the families and community members joining us today, thank you for tuning in. Again, thank you, Ash and Catherine and Dyne Therapeutics, for bringing all this information into our homes. For anyone, we do have a few more webinars this year. We have 5 more, so feel free to go to our website for more information. And if you have any questions for Dyne, Catherine's email is right here, catherine.beaverson@dynetx.com. Or you can always email me at Jett Foundation and we can get your questions, mara@jettfoundation.org. So without any further ado, thank you all again and hope to see you again soon.
Thank you so much.
All right, thanks for the opportunity.